Do it.
Hello.
Hello, everybody. How's it going? Welcome
Welcome
to
back,
another.
friends.
Welcome to another episode of the Rabbit Hole. I'm one of the hosts, Ben,
And I'm Ashley.
and this is a rabbit hole. Did we mention it was a rabbit hole? Anyway, today we want to talk about biology, and we can take it in multiple directions today. There's so many ways to look at it, from the smallest detail of inside of cells and molecules up to the large flora and fauna of biology and how it relates to our lives. So
Such a broad. I will preface this by saying that I do have a degree in cell and molecular biology. I worked very hard to get. So and then I worked as a scientist in the lab. So I have done a lot of biology, but I think the most important thing to admit with any kind of science is we don't know a lot. We do know a lot, but we also don't know a lot. So I will give the honest answers that I know, and when I don't know, I will say I don't know, because that's more important than saying or acting like I do know, because there's a lot we don't know, because cells are really, really complicated.
Yes.
And amazing and beautiful and intricate and unbelievable and magical, but complicated.
Yeah, I think for this podcast, I think it'd be good to do a little bit of an interview thing, because you have a lot of knowledge in this subject. You got a degree in this subject. You're passionate about the subject, and I am definitely more of the novice in search of answers over here. So we can just open it up and say, why do you like biology in the first place, and then why do you get excited about biology? And what makes it fun for you?
Oh, yeah. I think I really discovered how much, so I loved chemistry, and biology is a lot of chemistry. I think I really figured out how much I loved biology in my freshman Intro to Cell and Molecular Biology course.
Freshman in high school?
Freshman in college, sorry. I think I kind of knew, but that course that was the most in-depth I had really gone into a cell, diving into the world of a cell and learning all of these things. And then also just how much else we learned in terms of the extracellular matrix and cell transport. And then diving into, of course, cell respiration and metabolism, and then the cell cycle. And really kind of realizing it blew my mind. I remember sitting in Boston Hall at NC State, this is happening inside of all of our cells all the time. You get inside mine and my dogs and you and the trees, and it's just amazing, because I really came to appreciate that it's the air that I breathe out that fuels the plants, and it's what the plants release that fuels me. And I was like, this is literally the foundation and the why everything is so interconnected. And so I think for those reasons, and just it's you can't even really comprehend how small a cell is, and then how small all the molecules and organelles inside of it are. And just I think my brain just fixated on it, and I was like, oh my god, this is amazing. And just how much goes on. And I think all of the regulatory processes that cells have, such that it's kind of amazing that things do go wrong, but then it's also amazing that things can go right with how many steps there are. I think in that class, I really was like, okay, I need to do whatever it takes to get to biochemistry. That was the exact thought process I had in that class. I was like, however I get to biochem, that's the track I need to go to. Because I fell in love with the cell respiration unit. I wanted to know everything I could about the Krebs cycle, the citric acid cycle. And I want to know everything about membranes. I love membranes. Membranes are unbelievable. The fluid mosaic model, right? Eukaryotes are characterized by their membrane-bound organelles. And membranes are really, really fluid, and they're also really, really strict about what gets in and what goes out. And I was like, I just have to do whatever I can to learn about these, to learn about drugs and how drugs interact with cells. And so I really just kind of fixated almost. I was like, okay, I've got to take organic chemistry if I'm going to get to biochemistry. And I took orgo, which I absolutely loved. Love to even do an orgo podcast. We could talk about how reactions are driven forward. I don't know if you didn't take organic chemistry, did you?
I took regular schmegula standard
Okay.
chemistry.
Orgo was amazing. I loved it so much. I still love it. I still can remember a lot of it because I had such a great professor. But yeah, so I think I kind of in that. I think what hooked me about biology was more the cell and not so much. I didn't really fall in love as much with like the ecology. And don't get me wrong, that's still an amazing part of biology or evolution or biodiversity, but that's all still unbelievable. But for me, when I'm talking about biology, I'm more talking about cell biology, cell and molecular and developmental biology, I guess. And then developmental biology as a senior in college, when I took that, um,
So
my
what is developmental biology?
yeah, it's kind of how do you go from um, a single-celled organism or at the union of the egg and the sperm to an embryo and to then
Mhm.
a baby. Um, and what genes control what. And so we learned all about all the different genes that control and the developmental pathways of what forms this and what happens here and how do these cells, why do you get a nose, what splits the two eyes into two, like what forms the face, what creates the internal left and right asymmetry of the body, right? If you outside our left and right look the same but inside they're totally different, right? Like your left lung and right lung are nothing the same because the left side has to have room for the heart, right? So how the heart sits is different. And on the left side, you have different organs than on the right side. And so inside, you are totally not symmetrical between left and right. And so, what kind of genes are controlling to that? And if you knock out one gene, what happens to the rest? Like, how does one gene affect the rest of the cascade? We learned all about the different gene clusters and families that are so crucial for creating an organism that is biologically correct. And then also going through that. But what's amazing about developmental biology is because it's how something comes to be, more or less. And so then thinking about diseases as developmental biology. So thinking of cancer as a developmental biology pathway. What is going wrong or what genes are getting turned on or turned off that lead to this progression of cancer? And so that's kind of in a nutshell what developmental biology was, and looking at cell migration and looking at, how do cells decide, right? This thing called cell differentiation, which is when a cell decides what it's going to be. So all cells are genomically equivalent, meaning every single cell in our body contains the same DNA. And it's just where the cell is and the buddies around the cell and them talking to each other that tell the cell, you're a liver cell, you're a lung cell, you're a brain cell, you're an eye cell, you're an epithelial cell, whatever. And so learning about that, how do cells determine which pathway they go down? And then at what point is it 100% commit? What point can cells go back? What point is, past the point of no return? You can't go back. You will become, a kidney cell.
That's crazy. That is nuts. That doesn't make any sense. So I'm just thinking about, let's just say a baby, the formation between two cells that somehow creates, it has all the information. And that is crazy. That really is nuts. So
Yeah,
developmental biology sounds very fascinating.
it's
Was
unreal.
is and what was your major in? Was it in the, you said it was in developmental
It was cell molecular and developmental biology. Yeah. Yeah. So I focused on all of those in college.
cell biology, molecular biology, and developmental biology?
You got it.
Okay.
Those were my three areas. And it's unbelievable, right? Like how
That is crazy. Did you just like,
it fuses, and then everything that goes on before the two gametes fuse, right? Like before the egg and the sperm unite. How did the egg become the egg? How did the sperm become the sperm? All of the processes of meiosis, creating haploid cells for humans, for example, right? Our complete genome is made up of 46 chromosomes. You get 23 from mom and 23 from dad. And meiosis is the process of taking a cell and turning it into a haploid cell, meaning it only has one set of chromosomes. It only has the 23, so that 23 and 23 can fuse, and now you've got someone with 46, and they're going to start to develop. And looking at what's telling what organs to form, right? Because everything forms in a very regulated, function, like the way things start to the progression of how they start to form. And it's just unbelievable. I mean, I was just like, yeah, I have to know everything that I can about this. And so I was religious. I read the books. I was like, I will read everything that is assigned. I will watch every lecture I can. Like I just really wanted to understand. And I think it just like how could you not find it kind of miraculous? so
There's
so yeah, and then what goes wrong, and then anatomy and physiology. I took 2 semesters of that. So focusing on all the different systems, and I had my anatomy lab where we learned all of the muscles, all of the bones, all of the different, you know, um, insertion sites of muscles and origins of muscles and actions and agonists and antagonists. Agonists are muscles that help the muscles. Antagonists are muscles that work against the muscles. Um, and it's just yeah.
what is that? You have muscles that work against muscles?
Mhm. Yeah,
What,
that
what
oppose
is
it.
that?
So like muscles that keep a muscle from maybe hyper extending or from getting too long, you have to have some kind of opposition, right? Like you don't want to be able to do different things with your arm because then you would risk dislocating your arm or hurting your arm. So you're going to have things that oppose the motion so that doesn't happen. So
Oh,
it's this innate system of checks and balances within the body.
and that's more to do, I guess, with more anatomy and physiology. But I always thought that like what stopped your arm, I don't really know whatever. Well, I just thought it was kind of like your bone structure and like your tendons and ligaments kind of stopped from.
It all kind of works together, but you're having muscles that work as the antagonists, right? So like a big one to think about is the quadriceps, which are the top of the thigh, and the hamstrings, which are the back of the thigh. And those have a beautiful antagonistic relationship, right?
Okay, so it's more like it's not. I was thinking like there's in the same muscle you'd have antagonist and protagonist, but you're saying it's more like.
You can for muscles that have different, right? The biceps brachii are called biceps because they have 2 heads. Bi and triceps are called triceps because they have 3 heads. But they mostly work together. Yeah, it would be like the biceps and triceps opposing each other.
Okay, I was thinking like in the same muscle fiber. Okay, I got you.
No, it's this body's just amazingly intricate set of checks and balances to protect itself, and so that's why like you extend your leg and or at a certain point you're going to start feeling some kind I shouldn't do that anymore. It's your body being like, yeah, let's not do that because we're going to hyperextend and then we're going to risk injury. So the body is unbelievable at sending signals, and especially like I loved the endocrine system. I fell in love with bones because I was like, you think of bones as being these static, boring things, but there's so much going on in a bone. Right now, our bones are remodeling, they're reforming, responding to, signals in the blood. Like if we have a surplus of calcium in our blood, your bones are going to say, okay, let's reserve that calcium. Bones serve as a major storage site of calcium and phosphate. So your bones are going to say, well, we'll take that calcium up, and you're going to get osteoblasts, which are a specific type of bone cell that are going to come in and help add that calcium that's in your blood to your bones. Similarly, if we're in a calcium deficit, let's say, you're going to have osteoclasts, another type of bone cell that are going to come in and say, okay, let's chew up the bone so we can break down some bone and have some calcium to work with. You need calcium. Calcium is a crucial cofactor to drive a lot of biological reactions forward. So if you have a calcium deficit, your body says, we'll tap into bones. And then you can start getting bone breakdown, actually. So your bones are these really, really, really dynamic structures, and they have all this stuff that's going on inside of them. And yeah, bones are really, really, really cool.
Would you agree with the statement, if the bones are good, the rest doesn't matter?
No, because there's so much else at work here. I mean, that also comes down to like cellular communication. If the communication isn't good, if you have some kind of issue with a messenger somewhere, that's not good either. So not necessarily, but bones are really, really interesting in how kind of dynamic they are.
What's your favorite system of the body if you had to pick one? You have to pick one.
I can't.
You have to. It's just...
They all work together. It's just because we can't comprehend how intricately intertwined they are that we have to break them apart. But probably the endocrine system, the hormones.
Is that does seem to kind of go along with your cell molecular and developmental biology background, because it's hormones and like interacting with. Yeah, I could see that.
the hormones are amazing, and how regulated they are, and the different parts and ways that they originate, and like, and then the impacts that they have. Like, you have hormones that are made in the brain that are telling you, or the female specifically, like when to ovulate and when to start your period, when to start menstruating, and that all comes from hormones that are produced in your brain. And so those hormones that are in the brain have to somehow make it to the ovaries and tell them, hey, it's go time, let's go. Or the uterus, like, come on now. Or they work in conjunction with like estrogen and progesterone, which are the other two
Mhm.
players in female hormones.
I've heard a lot of that stuff only because I've watched there's a guy on YouTube called More Plates, More Dates. It's a classic. And the crazy part is they're like the biology, like cell molecular, like developmental, all these things, anatomy, physiology, they all are intricately woven together, just as each part of the biology stuff is woven together too. But you'll have weightlifters and like bodybuilders dabbling into trying to, they don't know exactly what's going on underneath the hood, but they're dabbling with these substances trying to get a desired result. And
Right.
they're willing to do trial and error on their own bodies, instead of knowing exactly what's going on, um, which I find is interesting because it's so complicated. It's like, well, let's just see if this works. And they talk about like different steroids, like I think it's like Tren cycle, and I'm not sure. There's a lot of different cycles, but they talk about, they're like, they sound like experts in endocrinology, because some of them do, because they're just tampering with everything all the time, and they'll like get the blood tests all the time, and like they'll go bald, and then they have reasons for that, because just trial and error of figuring
Yeah.
out when you start tampering with things, things can get it just shows how complicated we are. And I think it's fascinating that, even people that don't have degrees in biology, or don't have these, they're still, you can still use these things at your own risk, but to try to get a desired result by changing hormones to change your body. It's like crazy. It's like, it's insane. It really is. The endocrine system and how tweaking hormones can really affect your mood, affect your body.
Yeah. It
That's nuts. That's just nuts.
Hormones are these unbelievable messengers that travel great distances and have substantial impact. And I mean, you have hormones that impact all of the all of the organs in your body. Another big one is your way of salt and water balancing through the kidneys, and different hormones with that. You've got ADH, which is anti-diuretic hormone, which makes you retain water. So anti-diuretic, right? A diuretic is something that makes you pee. You're not doing that. And so when your body can tell that maybe its salt ratio is a little too high, you've got too much salt, you need some water to dilute it. It helps with the reabsorption of salt so that you're drawing more water into the body. That's how ADH works at the collecting duct, which is what ultimately runs down to the urethra so that you can, excrete excess water. But if you're not in a position to lose water, if your body is in water retention mode, you're going to upregulate that hormone, and that's going to affect the collecting duct and the permeability of the collecting duct in the kidney, so that you don't excrete water, and so that your body can return to homeostasis, which is biological, kind of normalcy, where everything is working together. And that's what we're always working on, homeostasis. So if you got too much of something, de-escalate. If you don't have enough, escalate. How can you do that?
Okay, so if the human body is incredible, cells and the cell systems and cell cycles are incredible, and the goal is to reach homeostasis.
Correct.
But humans and just really a lot of nature is extremely adaptable. So why does it really matter if your body's just gonna be fine regardless? Your body can adjust, right?
No, I mean, within a certain realm, yes. But if you it's like a seesaw. If you teeter a little too much, you're gonna start falling, and your body cannot. You only have so many checks and balances, and you only have so many security blankets, if you will. And your body is relying on how you fuel it in terms of exercise, movement, water, food, nutrition, sleeping. Like, your body is relying on these cues, and it's kind of like, if it takes enough blows, it is no longer able to function properly, because cells require proper nutrition and cells need something to work with. And if they don't have anything to work with, they can't do their job as well. And then if you're looking at over time, cells that are continually unable to do their job, such as immune cells, or if you're taking repeated hits to the gut through taking, obscene amounts of antibiotics, eventually all the things. So this is a huge thing to understand about the gut, is that the gut microbiome is our way of navigating all of the foods. Sorry, that totally scared me. Did you just see me jump? That caught me off guard. But the all of the gut microbiome is the body's way of navigating all of the foods that we're able to eat, because with all of the enzymes and all of the crazy compounds in foods, there's no way we would be able to digest all of those. And so what we've done is created these symbiotic relationships with microbes, which is the gut microbiome. And genetically, you and I and all the other humans are about 99 point something percent similar. We really differ very little in terms of our genome, and it's only in these different areas of the genome that's where we diverge. But our gut microbiome, you and I could have 0% overlap in our microbiome.
Whoa.
That's how diverse we are. You and I might not have any of the same colonies or strains of bacteria in our gut, or archaea, which are also able to, there are other single cellular organisms that are able to survive extreme environments. They're prokaryotes as well. And that's what's amazing about the gut microbiome. And that's why some people tolerate some foods and other people don't. It all depends on what have you got in your gut and how are those microbes working for you. And do you have microbes which produce short chain fatty acids, which are really healthy and are really good for you, or are you not producing those? So the gut, the gut is unbelievable. And that is a huge sense of variability and differences and divergences among us as humans. Obviously, you get a lot from mom when you're in utero. Mom is exposing you, and when you're breastfeeding especially, and if you're delivered naturally, you're exposed to so much throughout the vaginal canal. That's where the baby really starts getting introduced to this world of microbes. And then through breast milk, and then eventually the baby is going to start forming its own gut microbiome, which can be yet another. I'm sure you've heard of the gut-brain connection. And
serotonin levels are connected to the gut, and then
most of your serotonin is made in your gut. So it's about having a microbiome that works for you and not against you.
Okay, so
because if you have a bad one, you're in dysbiosis, which is any state of kind of imbalance.
So how do we change our gut biomes? What's a good gut biome? Can we tell the difference between a good gut biome and a bad one? Of course it's not, there's probably a lot of right answers, but the optimal amount of microbes in our gut that achieves the best way to digest food and absorb food that we intake.
Yeah, so I don't know if we've really gotten to a numerical kind of answer yet, but what we have determined and what has been repeatedly backed by science is the more plants you eat, the better. Diets that are heavy in meat and dairy are not conducive to a healthy gut microbiome. You're going to get microbes that are disease promoting, that we know are correlated with different diseases, such as type 2 diabetes, even colon cancer. And that's coming from diseases or from diets that are really heavy in meat and dairy. So the standard American diet, unfortunately. And that's why we're seeing a lot of these diseases. That's why we're seeing heart disease, atherosclerosis, building up of plaque along the arteries. And then those clots can eventually break off, and that can become a fatal event, right? Because the clot goes to the heart, and then the heart can effectively pump blood.
What about sugar?
Sugar is the sociopath of society. It says it's sweet, but it's poison. Sugar is really detrimental. It depends what type of sugar we're talking, though. Are we talking your good old table sugar?
Cane sugar, straight from
Yeah, that is bad sugar because it does not come packaged with anything else. So the sugar you find in fruit, fructose specifically, is packaged with all these other amazing nutrients and um, all these other phytochemicals that are going to help slow the release of sugar so that when you eat fruit, you're not seeing that huge spike in your blood sugar, your blood glucose, and then that's calling on insulin. And this is how you lead to a cycle that perpetuates insulin resistance, which is the precursor to diabetes.
I've heard this before, but does your gut biome cause you to have cravings?
It can, yes. Yeah, your bugs in your gut know what they need to be fed. And if you're looking at a healthy gut, it wants to be fed plants because that's how those thrive and that's how those survive.
Mm.
If you're got a gut microbiome that feeds on sugar and bad fats and other unhealthy greases, then that's what you're going to crave. So absolutely. But what's amazing is the gut microbiome is very resilient. It is very dynamic and it is very willing to change because there's so much bacteria in what
That's a good thing for people that have a bad gut biome.
It's amazing. We've done studies and we've seen that biomes can change in as little as 21 to 28 days.
Now, so
We're not talking a year-long process. We're talking if you clean up your diet or even just starting new plants. Now, the thing is, a lot of people say, like, I don't want to eat beans because they make me gassy, or I don't want to do this because it causes me discomfort. That's because you haven't built up the microbiome to handle it yet. That's why a lot of people struggle with raw food. Raw food is very hard on the digestive system. I'm specifically thinking raw greens like kale, for example.
Broccoli.
Yeah, it's hard on the digestive system.
It's hard on your jaw to eat raw broccoli. Like,
man, I was eating a kale salad the other day and I was like, holy goodness, I'm getting a jaw workout if that's even a thing. I'm gonna have crazy defined jaw. But it's hard because they're so fibrous. And so if you don't, so here's the thing. We talk about a lot of nutritional deficiencies, but one of the biggest nutrients we are lacking in our diets is fiber. We hardly eat enough fiber because we're not eating a lot of plants. Our meals are not plant predominant. I'm not even saying you have to adopt a plant exclusive diet, and I'm not saying that at all, but I'm saying our goal is to eat as many plants as we can, because all of those plants are packed with different nutrients and different vitamins and different things that are so nourishing for us. And then those are going to start colonizing different bacteria that are going to work with us. So if you can't handle beans or raw greens, it's because you just don't have the digestion for it, because you have microbes that don't know what to do with it. And then that leads to discomfort. But if you have microbes that are like, yes, kale, yum, right? And they're like churning it out for you, and then you're going to get all these amazing things that the microbes start producing, which are crucial to a healthy system and a healthy body. But your gut, I mean, the thing is, a lot of us have such damaged guts because of what we put in our mouth multiple times a day, because of stress. I mean, a big thing that also contributes to gut is stress. Certain microbes just can't live in a stressed environment.
The cortisol kills it or something like that.
Yeah. You're just unable to thrive. So if you're because of all the stress and anxiety, right? Because of what the anxiety response sends your body into, you're now in this fight or flight systemic response. You've got adrenaline pumping, you've got cortisol that's taking a hit to the microbiomes. It's slowing digestion down, right? When you're in fight or flight, your body says, I'm only sending resources where I need them. I need to be alert, right? That's why you get hyperactive, you get hyper responsive. And it says we've got to slow digestion down, we've got to slow the excretory system down, we've got to ramp up the immune system, because we've got to be ready to take an injury, right? Our body thinks not fight or flight of, oh, I've got to go talk to my boss. Our body thinks, oh, shoot, some kind of animal is coming after me, or I'm on the run for my life. And so the fight or flight response is really powerful for those things, but now we tap into it for so many events that aren't really fight or flight, but our body hasn't really made that distinction yet in terms of, oh, my life isn't on the line, this is just a stressor, but you still go into that overdrive. And so then all of your body takes a hit and you get this ramped up immune system, and then that leads to chronic inflammation.
So it sounds like if you're in, let's say somebody's in survival mode, where they're in a constant stressed environment, it's almost like a vicious cycle. Maybe it is a vicious cycle to where they're stressed out, and then their body is not letting them their body is spending all the resources trying to stay hyper alert for fight or flight, instead of putting more of the resources towards the digestive system to where they can actually absorb nutrients, which would maybe allow the person to get the energy that they need. But the cells are burning stuff so fast, it just sounds complicated. And I think that's one thing, just from hearing you talk, it sounds like not only is there beauty in the complexities of biology, but also this complexity and being able to see when things go wrong and how things work provides an understanding that points to nutrition, exercise, sleep, healthy lifestyle, because we all know these simple things. Like, we all know eat more vegetables, we all know sleep more, we all know to exercise. But why do people not do it? It's because a lot of times they don't understand how important things are. They don't really understand it. They've just been told it. And this type of knowledge that you're saying is one of the many avenues to reach some of these conclusions about more healthy lifestyles and the way to take care of your body. And that is very fascinating to me, that biology is one of the many avenues to reach some of the same conclusions that you can reach in other areas of life.
And I think what's amazing about it too is, like, this isn't even, ancient wisdom, which it is in a lot of ways, right? Our ancestors knew. They were walking 10, 12, 15 miles a day. They were growing their own vegetables. They were getting exposed to so many microbes because they were pulling stuff right out of the ground and eating it. And it's amazing. In the handful of soil, it's something crazy. Like, there are more species in that handful of soil than there are seats in the PNC arena. Just in a handful of soil. Like, it's amazing all the microbes that there are, and we don't even know about
We need to start
several.
eating dirt.
Well, dirt
A
has some.
little bit. Stop washing our hands.
A lot of people say, like, with gardening, and that's what I try to do when I garden, is that exposure to the soil through your hands and all that dirt is really important. I mean, unfortunately, the soil that we're dealing with isn't the soil from 200-ish years ago, before the Industrial Revolution and before we started releasing all these chemicals into the earth and into the soil. But it's amazing. Yeah, biology is evidence-based reasoning why these are the things we should be doing, which a lot of us already know. Like, you eat plants and you're like, I feel awesome. Maybe your body has to have a few days to adjust, but and a lot of health experts agree on this, is that beans are some of the most protective foods you can eat. If you're gonna up anything in your diet, make it beans.
Should
So
you go easy though, to kind of ease your gut biome into it if you haven't eaten beans in a while?
Yeah, you might need to ease your way into it, because beans are really fiber dense. And so you might have a microbiome that's like, whoa, I don't know what this is, and I don't know how to handle it. That's okay. You just have to increase your exposure to it, because you will get there. Those bacteria will start to colonize, right? Your gut is ultimately a reflection of what you eat. And so the biggest thing to take away, though, is that we have moved to this we have shifted to this, the predominant food on the plate is a piece of meat, and it's no longer the vegetables. And so even if you're like, I'm not giving up meat, that's okay. You don't have to. Nobody's telling you that's what you have to do. What everyone and what the true information is just get more plants, whatever that looks like. And I personally I strive to get at least 20 different plants in a day.
Wow.
Which it takes a lot of planning. And I mean, it's a transition. Like, you're not going to do this overnight. This has taken me a couple years to figure out, to navigate my way through. But when you do it right, it's not that challenging.
I have a question for you.
Okay.
So in jiu-jitsu and these grappling arts, there's always people. It's very common to get skin infections such as impetigo or ringworm or even MRSA.
Sure.
And so what people do is they take antibiotics, and because that's one of the best ways to get rid of it, especially if it's like MRSA or something.
Yeah.
It's just flesh-eating bacteria.
Totally.
So,
yeah.
So after hearing that, it sounds like to me that not only are there microbes only in your gut, they're probably everywhere in your body, right?
Your skin is covered in them.
Okay.
You have so many bacteria on your skin.
Do we have microbes in our like lymphatic system, in our brain? Does that stuff?
Not really. The brain is super protected by the blood-brain barrier.
Okay.
But any kind of orifice or opening on the body, you've got microbes there.
Okay. Now, what happens if I take probiotics and antibiotics at the same time? Who wins?
That's an awesome question. So antibiotics, I'm definitely not saying that they're bad because I think they are the single most probably the number one discovery in medicine because of the lives that they've saved.
And then when do you take them too? So
Well, I think that that's the thing that we're seeing is that there's a lot of antibiotic over prescribing and that a lot of people are just taking antibiotics kind of when they might not be necessary. Now for something that it's like MRSA, I think that's totally appropriate. But a lot of people might have something viral going on and they're giving them an antibiotic, which obviously isn't going to help, right? Viruses aren't bacteria, so they won't be receptive to antibiotics. That's why you hear the viral, oh, it's just got to run its course, and that's what has to happen. So there's actually an interesting study that's pretty recent that showed that if you take an antibiotic, so antibiotics are not specific, right? What's amazing about antibiotics and why a lot of them work is because ribosomes, which bacteria have ribosomes. Every living cell has a ribosome because ribosomes are where the RNA goes to be translated into a protein. Ribosomes are the protein builders of the cell, okay? And you have to have protein to survive because proteins do basically everything. They are like the workhorses of biology, of cell
and there's
biology.
different types of proteins, and then different type of because of the different types of amino acids that make it up.
Right. It's amazing. Like, okay, right. Think about the English or the English alphabet and how many words you can create because of it. It's the same idea with proteins. You've got 20 amino acids, and then there are infinite possibilities of.
And proteins are made up of as many or as little amino acids. Like one protein could be made up of 40 or 50 or is it like hundreds of amino acids all combined?
Yeah, exactly. So proteins are really, really interesting. So what I was going to say real fast though is that ribosomes have two subunits. Okay, they have one subunit and another subunit. And what happens is when a ribosome forms, the two subunits join. Okay. What's different? Why antibiotics work for the most part is because eukaryotes, which our cells are, and prokaryotes, which bacterial cells are, have different subunits of the ribosome. So antibiotics target the prokaryotic ribosome. And then that makes the cell unable to produce its protein.
And then
and so it falls apart, right? The whole machine falls apart.
Okay.
So that's how a lot of antibiotics work, and that's why they work selectively. That's why they don't hurt our eukaryotic cells, but they do hurt the bacteria.
And there are different types of antibiotics.
Absolutely, yes.
Even though they're all prokaryotes that they're attacking, there are different types of prokaryotes, and each antibiotic is geared specifically towards certain ribosome.
Yeah, and there are other things too. It could be cell membrane stuff, right? So they could be doing things to the cell membrane and poking holes in it, which then the cell starts basically bleeding out, if you will. But yeah, different antibiotics work better against different strains. So antibiotics for that reason are not selective. It's not like you give an antibiotic for a specific strain, and that is the only strain that takes a hit. Everything else takes a hit too. And so if you're going to take antibiotics, you need to make sure that you're still giving yourself those quality prebiotics and probiotics. However, this study, and that was what we thought. This study came out though that actually showed that probiotics can, taken after a cycle of antibiotics, can actually interfere with the new colonization of different strains. So that just came out, I think, like maybe a month ago. So I think now there's a little bit of like, whoa, going on in the scientific community about what is it. But probiotics aren't going to save you. They're not lifesavers. That's your diet.
My kombucha is going to save me. How about that?
Okay. Then we'll do case study. I'll take you with me to the trial.
So,
but your diet is your number one line of defense.
Okay. what is that quote that's in your kitchen?
Uh, which one? Oh, let food be thy medicine.
Yeah, I like
Yeah,
that a lot.
Hippocrates. Let food be thy medicine. And that's not to knock modern medicine at all. Like there are obviously instances where modern medicine is amazing and crucial and life-saving, and thank God we have it, right? But I think like it's this other kind of just perpetual cycle of we want a quick fix. And for optimal health, there isn't a quick fix. It's
Yeah,
what your day-to-day habits are.
yeah, I think that's the main draw is like that's how sales works. You buy something and then it's solved. It's a one-time
Right.
decision that you only have to make once, and then your problem is taken care of. But in reality, in life, a lot of times it's a daily decision, and maybe even multiple times a day decision until the habit gets ingrained. But I do want to talk a little bit more, just because personally, it just seems very practical about just with like jujitsu community, and there's like antibiotics. And I know there's Lotrimin, which is a topical cream that you can put on stuff. And so does that cream have antibiotics in it that kills the bacteria or the microbes, or and then
I'm
how does taking something orally end up like the way the medicine is transferred when you take it orally, somehow that ends up taking care of your skin and your gut biome on accident, and the complications that come with that. Like there's one guy, he's the best in the world. Um, he took too many antibiotics because he kept getting staph infections.
Yeah. Yep.
And so he was wrecked. His body was absolutely, he could not eat. He was nauseous 24/7. His name's Gordon Ryan, and he had to stop doing jujitsu. And uh,
Um,
he ended up reaching out, and they found something with his gut, but his gut got thrown off, and it just caused a cascading events. And so that makes me think, like, um, just I feel like it's tough with these antibiotics. They just take out everything. But I don't know. It just seems like a weird thing. And prebiotics, probiotics, antibiotics, and how to navigate that, especially with these like trying to target them.
it's really confusing, and that's why I'm hoping to go into this field of work, because this is what I really drive or derive a lot of joy and pleasure from. So I'm definitely not an expert. But I think Lotrimin is an antifungal. Yeah, I'm pretty sure it's an antifungal. So fungus is different than bacteria, right? Fungi are eukaryotes, so they are more in line with an animal cell than a plant than a bacteria, because fungi are eukaryotic organisms,
Mm.
which is interesting. We think of them as prokaryotes, but they're actually eukaryotes, so they have membrane bound organelles. Um, but right on your question, I think that what we're seeing with all this antibiotic overuse the most is these superbugs or antibiotic resistance. Now things that antibiotics used to be able to treat, we're no longer able to treat. And actually, something that's cool about antibiotics is most of the antibiotics that are manufactured are going to livestock, and they're consuming it before slaughter.
Oh, so then it gets passed down the chain.
So then, right, you were innately, like, just inherently consuming antibiotics by eating meat. And that's where a lot of antibiotic resistance is believed to be coming from, because we're getting exposed to these. And then bacteria are not dumb, right? We saw this with the COVID virus, even how it kept mutating, right? We had the Delta, and then we had Omicron, and we have different variants. Bacteria are very similar. They will find ways, right? It only takes one bug to survive of a species, and then it starts replicating. And then all of a sudden, and bacteria can, in a matter of 20 minutes, colonize something. Bacteria have a crazy fast reproduction rate. And now you've got a bunch of bugs that are super resistant, and are not going to be knocked out by that antibiotic. And now they're no longer susceptible to that antibiotic. You have that antibiotic resistance. So this is how a lot of molecular biology works. When I'm cloning something, when I would clone something in the lab, I have an antibiotic resistance gene. So let's say it's a kanamycin, which is a type of antibiotic. I have that resistance gene in my plasmid, which is a circular piece of DNA that I've transduced into a cell to contain that plasmid. So it now has that bacterial DNA. Now what I'm going to do is I add kanamycin to my plate, and only the cells that have the kanamycin resistance gene, which is in line with the other gene that I want it to take up, will survive. Does that make sense?
Uh, Not exactly.
Okay, so a plasmid is a circular piece of DNA, right? So our DNA is linear. We
Okay.
have the double helix. A circular piece of DNA is called a plasmid. So within that plasmid of a bacteria, you have an antibiotic resistance gene.
So our DNA is linear.
Correct.
Bacteria's DNA is circular.
Good. Yep. So when you have a plasmid, and this is what you can do, you make these plasmids that have a gene of interest. So like, let's say I want to get a gene into my cells to study. I'm going to put that gene into a plasmid that has a specific antibiotic resistance. So then the cell, just imagine it kind of eats this up, this enters the cell, and now this plasmid is inside my cell. Okay.
Also, bacteria go inside, they go through your cell membrane.
This is isolated DNA. This is isolated DNA from a bacteria. Bacteria themselves, no, unless you have something wrong with the membrane, they should not be able to penetrate a cell. I'm saying if I've isolated a piece of DNA from bacteria, if I've put the cell through enough cycles, right, of kind of breaking it open, lysing it, and then all the DNA comes out, and then I keep binding the DNA to things and eventually elute it, and I'm left with my DNA.
Okay.
Are you with me on that?
Yeah, I didn't know that you could break down cells like that.
Yeah, you lyse cells. This is what I used to do all the time. You lyse cells, burst them open.
You're saying lyse?
Lyse, Lyse. Lyse. Lyse is a common word in biology to break apart, like hydrolysis, hydrolysis, breaking apart with water.
Gotcha. So you have a bacteria and a cell of a bacteria, and somehow you lyse the bacteria.
Yep,
and extracting the plasmid, which is the DNA inside of the cell.
Perfect.
And with this DNA inside of the cell, you are putting it into your DNA or into your cell.
Yeah. So this is what can happen. Okay, so I have my circular piece of DNA. I can use different enzymes to break it apart. Okay, so different enzymes can come in at different points in the cell and break it apart.
And you've broken up the plasmid, which is the DNA of the bacteria. So
yeah. Yes, and I do that with things called restriction enzymes, which find specific base pairs. They're kind of like barcode scanners. It has a specific barcode, and it knows, oh, here I break the DNA.
Okay.
So it comes through and it literally snips the DNA. You cut the DNA. So now you're left with a plasmid that's no longer linear.
No longer circular?
Or circular, yes, excuse me.
So now it's kind of linear. It's linear.
Yeah, okay. Exactly.
Gotcha.
Now what I can do is I can say, okay, here's a gene that I'm interested in studying, and I can put that gene kind of like copy and paste.
A gene in the bacteria that you're interested in, the DNA of the bacteria that you're interested in studying. So you're saying, I'm going to study the bacteria of the DNA inside of this bacteria, just a part of it, and I want to do what with this bacteria's part of this bacteria's DNA?
So I'm using the bacteria DNA, the plasmid, because I'm going to make it a plasmid again. I'm using it as a vector to put it into a cell that I'm interested in studying it in. So I'm going to take this bacterial piece of DNA with my gene of interest and make it go into a human cell.
Okay.
HEK cells, human embryonic kidney cells, are really common ones to use. They're like some of the workhorses of cell and molecular biology. So I have my plasmid, and then I've cut out a piece of the plasmid, right? I've cut out part of the DNA with these restriction enzymes that's snipped. Now I take my gene of interest that I've isolated and I put it back in. So I basically flip-flop them.
You put it back into what?
Into the plasmid.
So you took it out, you moved it over to the hex cell, which is a workhorse cell.
Not yet. I haven't done that yet. I still have my plasmid like this.
Okay.
I still have my plasmid like this, and it's missing this part of DNA that was binding it, right?
Okay.
I put my gene of interest there, and because of specific enzymes and how the plasmid overhangs, it's going to anneal back together. So now the plasmid contains my gene of interest.
So you took out a piece
and put in the piece I'm interested in studying.
And you put in a piece from your own, from a human or the hex cell, whatever, into the bacteria.
Whatever I'm interested in studying, exactly.
Okay, now how does that, what's the goal? How can you study something? What are you looking for when you do this?
So I have this now, right? That I put the gene that I'm actually interested in now.
Yep.
Now I'm going to take this, and with transduction methods, which you can use different things. You can use electroporation, which basically you use high electric currents to poke holes in the cell membrane. And so this piece of DNA will just slide into your cell. Or you can use a lipid-based, which is a fat thing, and it kind of it surrounds the DNA so that it becomes nonpolar, right? Because the cell membrane is dealing with polarity, and we're all about polar gradients. And you make it nonpolar so that it'll go into the cell through the cell membrane.
And it go back into the bacteria cell?
Nope, now we're going into a human cell.
Okay.
So the whole point of it, and why I kind of took us down this rabbit hole, is that I've got this plasmid now, but what's crucial about this is this bacteria contains an antibiotic resistance. The bacteria that has my gene of interest, this plasmid that now I'm putting into a human cell, it has my bacteria, or my antibiotic resistance. So now on a plate that
you already knew that. You already
right,
knew,
I already know that. Yes.
you already knew what part to take out. You already knew what part to put back in based off of past research and knowledge. Right. And so you're taking it and putting it back into the human cell to see what happens.
To watch its effect, right? Or to do some kind of manipulations with it. Now what happens if I tell it to overexpress? What happens if I silence it? How does that affect these cells? That's what I'm going to be doing. But what's interesting about this is that antibiotic resistance acts as a selectable marker, is what it's called. So think about it. Any cell that takes up this plasmid, right, with my gene of interest, any human hex cell, let's say, that takes up this plasmid is going to be antibiotic resistant now. Because now this has been integrated into the cell. So then what I do is I blast my plate with my antibiotic. Let's say it's kanamycin, or it could be blastocidin or pyromycin or whatever. Yeah, it could be any of those. And then any cell that did not take up my selectable marker will die, because they're not antibiotic resistant, because they don't have that piece of DNA. So now I've killed off cells that don't have it, right? I've killed off any cells without it. And so now I know I'm working with a population that's only expressing what I'm interested in. It's kind of a wacky concept, but the only reason I was mentioning it was talking about what we've been able to kind of move forward with molecular biotechnology because of this antibiotic resistance.
So, and I imagine maybe the goals are different, but is the goal to study what happens, not necessarily to make things antibiotic, or
right?
Or it's the goal is not to make things antibiotic. It's to study them.
It's just this way to eliminate any cells that don't have what we're interested in.
Okay.
Because if a cell hasn't taken up this piece of DNA, right? If it hasn't taken up the plasmid, it's not going to be antibiotic resistant, and therefore it will die in the presence of the antibiotic. However, the cells that do have it will be antibiotic resistant. So after a couple days of what's called selection, where the cells that aren't going to survive will die because of the antibiotic, because they don't have the resistance, after that time, you will have a basically pure population of cells that have what you're interested in studying.
So you're saying antibiotics kill human cells?
Yes.
They do. They don't just kill the bacterial cells, they also kill this.
Yeah, that's why we're more dealing with like pyromycin and blastocidin, which are, from my understanding, more like laboratory antibiotics, because they're super potent.
So I guess people are using antibiotics in slightly different ways. One is maybe more for research of kind of honing in on what you need,
right?
And versus the ones that are trying to actually take out bacteria.
Right. And not all antibiotics just target, right? Antibiotics have lots of different ways of working. They have lots of different mechanisms.
Man, this is much more complex than I thought antibiotic was. Like,
Oh yeah, it's
Sure, I'm sure you're just scratching the surface too, and it's hard to, yeah, it definitely, I can see how it takes a degree to wrap your head around it.
and even still, like, I don't know that much. I mean, I know a lot, but that's I know a lot, which is a little, you know, and I'm okay saying that because it's true. Because these interactions, and I think, the interactions among cells, among molecules, it's so infinite. Like, you can't even really comprehend. But a big takeaway point from this is that a lot of the antibiotics we consume through the meat and the dairy that we eat, because the antibiotics get into the cow's milk, and then it's in the cheese, and it's in all these things. So if you're eating a standard American diet from, kind of like the normal stuff you would buy at the store, you're actually consuming quite a few antibiotics, and then that's killing off naturally just good bacteria in your gut,
Mhm.
which is something a lot of people don't realize. And of course not. Like, how would you know that? It's not like the food companies are promoting that information.
Yeah, and
So
I think that's why that's good about McDonald's, is because it's like it's not even meat, so you're probably getting something like super clean and like very quality tested, right?
I'm sure the quality is just through the roof. Go to McDonald's.
What about
God, have you seen that documentary that guy did where he took like some foods from
Supersize Me?
McDonald's? Super Size Me, I think is it?
Yeah.
And he just like grows them, and they're still growing like 30 years later, and there's no fungus or anything on it. God, it's so nasty.
Oh, that's not Supersize. Supersize Me is the one where he eats McDonald's for a month.
But yeah, that's it. But I think he still has, like, he just bought a quarter pounder or something, and he put it in a jar to see what would happen to it over time. And there's still like no sign of decay.
That's just uh built, it's American made, built to last. You know, that's good.
Well, like I said, the body can only take so many blows before it's like, okay, I can't fight anymore. And that's the thing, like cancer, you and I right now probably have 700 or 800 cancer cells floating around our bodies, which, given how many cells we have, is a pretty inconsequential amount. But our immune systems should be able to tolerate that, right? Your immune systems, you have these thing called natural killer cells, and they're in the immune system, and they're going to go around and they're going to eat those up. They're going to say, nope, you don't get to live. You don't make it, because cells have all of these intricate and unbelievable processes of before they move forward in the cell cycle, right? So the cell cycle is mostly made of interphase, but sometimes you actually do get into mitosis, which is when the cell actually divides, when the cell actually replicates,
What's
right?
the difference between mitosis and interphase?
They're in between. Interphase is one, and then mitosis is another. But interphase is like, if you think of the cell cycle as a day, interphase is from midnight to 11:59 p.m. and mitosis is only 1 minute.
Yeah. Okay. I got you.
Most of the cells spend most of their time in interphase, which is just G0, which is kind of an inert, just the cells alive hanging out phase. G1, which is the growth phase. That's where the cell starts growing, getting ready to divide. It says, okay, here we go. We're going to get ready. And so the cell starts growing, it starts acquiring all the nutrients it needs, right? It's really not an easy task for a cell to divide. And there are all these checkpoints that the cell has to go through. There are tons of regulatory standpoints, kind of like guards that are saying, yes, no, you, not you, yes, okay, fine, sure, go back, no, repair, fix. Like, it's this really well orchestrated and regulated process. And so if a cell is okay, then it's going to enter this S phase, which is the synthesis phase, which is where the cell actually replicates its DNA. Now, that is super crazy because it happens insanely fast in terms of how fast the machinery works. But then before the cell can move on, you're doing DNA repair, you're making sure that you have coded the right thing, you're making sure that nothing's wrong with the DNA, right? Because, okay, so this brings us kind of to the central dogma of molecular biology, where you start at replication. Okay, so that's the DNA just literally makes the copy of itself. That's all replication is. You make a copy of yourself. Then you go to transcription, where you transcribe. You take your DNA and you go from one language to another. You go from DNA to RNA. So from deoxyribonucleic acid to ribonucleic acid. And there are a couple key differences between DNA and RNA. DNA is double-stranded, and the strands are held together by hydrogen bonds between the base pairs. And RNA is just single-stranded. So you only have one strand of RNA. That's one of the big differences. Another is to do with kind of the actual molecular and chemical structure of it. So DNA goes to RNA. And then that RNA, that transcription happens in the nucleus. Okay, so you get this you get DNA that becomes mRNA. Then mRNA leaves the nucleus and goes into the cytosol, which is just the jelly-like substance of the cell, and it finds the ribosome, where it's translated into a protein. That's translation. And proteins, right? You have your primary structure of the protein, which is just the string of pearls, all the different amino acids. And that's just, 3 codons. So you have different base pairs, and the base pairs become, they're coded in triplicate. And so each triplicate is a codon, and it codes for a specific amino acid. So you have AUG that codes for methionine, and that's the start codon. There's this amazing effect called the wobble effect, where the last pair of the amino acid usually, so like if you have AT, or I can't think of one exactly, but like CAG, and you might be able to wobble that last base pair, that G, and still get the same amino acid. And that's kind of a really sneaky way of saying, okay, even if there's a mistake, this is still another check and balance that you're still going to get this this amino acid coded, and it's not going to affect it, right? Because if you start getting amino acid deletions or replacements, if you're getting a non-polar amino acid replaced with a polar amino acid, that's going to vastly change the structure of the protein.
So these biological mechanisms have several points of checks and balances. They have redundancies to where if something fails, then that's something else.
They have redundancy. That's what the wobble effect is. It says, okay, even if you accidentally code this as a C instead of a G, you're still going to get the same one. So it's really amazing. And it's amazing and miraculous how this happens and how this is happening. So then you have this one-dimensional protein, okay, that is just a string of beads on a string of pearls, right? It's just everything is kind of its own. And then you're going to start getting the protein to fold. And it's going to start kind of folding over on itself, and you start forming these things called alpha helices and beta sheets.
And this is all happening within the cell during transcription.
Translation. Yep. It's all happening. And then you start folding, and it folds over on itself. And then you get to this 3-dimensional structure, which is the protein. And that's where we bring in like crystallography to start kind of like trying to actually image the proteins. And then the protein, then it's not done. It's still a baby. It's got to go through the endoplasmic reticulum.
Of course.
Like, we got to make sure. And it goes through, and it's going through all these checks and balances. Does this work? Is this okay? Yes, no. If they say, oh, this protein folded wrong, it gets flagged, and it gets booted, and it gets degraded.
Man.
Chomp, 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 chomp. Not a good protein. Not okay. Not going to use it. Go get degraded.
I didn't know that there were so many redundancies and so many checks and balances in this. I just thought it was like mainly was just hit it perfect, and then the cells that messed up got eaten by something else. So
no. And cells, there are lots of ways. Sometimes, I don't know if you've heard of apoptosis, but that's basically cell suicide. That's when a cell says, oh, something's really screwed up here. I'm going to die. Dang. And it goes through a bunch of processes to basically kill itself, because it knows that it's a bad egg, or because it's reached the end of its life. Like some cells they just have short lifespans, and then the cell's like, all right, I've lived my life. I'm done.
What cells last the longest?
Your neurons.
Neurons.
Your neurons, yeah, because overall neurons don't replicate and divide. So neurons are really crucial. So that's why things like binge drinking or things like that can kill neurons, you're never going to get those back. So you've got to be really protective. And I would love to go into like neuron biology another time and talk all about action potential and how signals are communicated across the body, if you would be interested. But how it's unbelievable how they integrate all of this information and then decide if we go or if we don't go. And it's an all or nothing. And then how they release the signals into the synaptic cleft. I mean, it's just like all of this even is just barely at the surface. We're not even at the surface. We're like hovering above it.
Mm.
And it just is.
That sounds great for some other podcasts. I feel like, I mean, with pretty much any topic, as is the name of this podcast, you can just go down as deep as you like. And this is a fascinating subject that has real world implications. I feel like when uh, being equipped with this knowledge forces you, forces me to take things like nutrition, exercise, health, sleep much more seriously because um, it's hard to hide behind the evidence and like the consequences of not doing certain things. And
Yeah.
of course, everything's interconnected, but this is just such a good uh, perspective and lens to look at um, health through and just life through in general. Um,
And we haven't even really gone into the nutrition, like all of the different
True.
aspects of that, which just reinforces it. And that's what I said. I mean, that's not just it's to feel better, it's to be your best self. Like partially, yeah, but this is evidence-based reasoning and an evidence-based lifestyle and diet. And once you have the facts, it's kind of irrefutable.
hmm Yeah.
So I know this was like super information dense, so I don't want to overwhelm because I could keep talking all night because I just think this is so fascinating, and we didn't even get into like cell metabolism, which is another thing I
Yeah,
would
I
love.
was writing down a lot of stuff, and it's just it is so much. It's all and it's all seems pretty good. I feel like we could spend a whole podcast on each of them, and there's like 30 of them. You know. Um,
2022, the year of biology.
yeah. But on that note, my brain is getting a little tired. I'll
A
say.
little, it's a lot to think about. I know.
Yeah. Um,
So, but that was really great. Thanks for tuning in and for letting me talk and share.
yeah. And thanks for joining us on the rabbit hole, you listeners out there. Goodbye. Farewell.
Take care.